I do not have a very strong background in EEG. It was my goal with my postdoctoral fellowship here at BCBL to develop my skills in EEG. But I came in with the knowledge of pupillometry, and we actually have an iLink down in EEG cabin 1 that all the cords were run, everything was set up, it was working, but no one was running experiments with the two of them together. So my task was to figure out how to make PsychoPi and iLink and our EEG system all talk to each other. So this builds on practical example 2. So if you remember practical example 2, I showed you my PsychoPi experiment that talks to the eye tracker. It's very, very code heavy, but I did show us that we can use not experiment builder, PsychoPi's builder view to use a graphic user interface to pull together our experiment, click to generate code, and then we can steal code chunks we need and put them into this sample experiment that I've created that uses PsychoPi Coder, which is essentially Python code. The benefits here, it's highly flexible, reproducible, and free. It will require some knowledge of coder view. You can always go back and rewatch practical example 2, and I'll point out the critical code chunks that you would need to edit. There's lots of code for making PsychoPi, or just making the computer talk to the eye tracker. And what we'll see in a moment here is making it talk to the EEG system requires substantially less code, actually. So if you develop an experiment that has PsychoPi talking to the iLink, adding this additional step of making it also send what are called triggers to the EEG system is actually incredibly easy. I was shocked by how easy it was and also very grateful by how easy it was. So compared to the iLink system, our EEG system is pretty easy to talk to with PsychoPi. So there are sample experiments for you in the Google Drive. Once again, I actually haven't uploaded my EAT experiment. This is the one that I'm currently collecting pilot data on. So I wanted to check some things before I upload that. So maybe in the next couple of weeks, you'll see that get uploaded. The bare bones example is a lot like what I went over in practical example 2, but now I have also made it talk to the EEG. I'm sending triggers, which is that's the critical timing information that we need to extract data from the EEG system. So this is a very basic, that's why it's called bare bones, sample experiment where the participant is going to hear a stimulus each trial, have to wait, and then the cue on screen changes from a circle to a fixation cross. And they are supposed to repeat aloud. I do have another walkthrough written for this. It's another Word document. I know it's terribly boring, but I do go through in detail every line number where the critical information is. And so it's basically the same thing, except I have marked in orange where the EEG stuff is in this walkthrough. And now we can look at the code, and we're building on practical example 2. So I'm assuming some amount of familiarity. I don't expect you to have become an expert coder with Python or PsychoPi, but there are only 2 key code chunks that we need to talk to the EEG, which is pretty remarkable compared to how many code chunks we need to talk to the iLink. It's pretty great. So the first of these, you need to search lab hackers. Actually, it's technically 3 code chunks, but the first of these is we are loading essentially a package called lab hackers, and this is what we're going to call the function from. The second, you need to search p_port in the sample experiment. This isn't a name that is necessarily standardized. So if you're looking at other people's experiments, it might not be the thing you need to look for, but in the sample experiment, if you search p_port, it'll take you to any critical code chunk that has EEG. So you can see I have a comment here. This is where we establish our connection to the EEG system. So it is just like, okay, we are now talking to each other. The next time p_port comes up is within our routine. So this may or may not be reminiscent of session 2, but at the start of a routine, you can see there's some code that's really typical for PsychoPi, like has the routine been told to continue? Has it ended yet? Not yet. And it's setting up for the routine, and it's keeping track of what components there are. P-port becomes a component in the routine. So this is just a critical thing. Your routine could have many things. My routine only has trial_audio because this is a super simple experiment. If you're presenting 6 images, all of your image components are going to be listed here with comma separation. But P-port needs to be here as well, because what this is going to do is it's going to keep track of that component. All the components in this list will be kept track of. If we search for P-port again, we get to the meat and potatoes of it. This is where within our routine, we can see we have some stuff about trial audio starting. So this is our routine is playing the audio. That's the main job of this experiment. Below it, we have P-port updates. This is very standard PsychoPy formatting. When you generate code for any kind of component, it sort of looks like this. It's keeping track of whether a component has started. And in this case, I have told this component not to start until the trial audio starts, because I want it to be perfectly lined up with when that audio stimulus starts. This is my timing information for when I go to analyze the EEG data. Down here, this is telling this 0.5, this number is telling this trigger to be sent for 500 milliseconds. Whether you can go shorter or longer, I haven't really investigated, but this is just what I had seen in another experiment. So it sends this trigger, and long story short, you can do both at the same time. I am still working on this experiment. So I have collected one pilot person. I need to actually look at the data and, make sure everything is going as smoothly as I certainly hope it is. But we have a room downstairs. EEG cabin 1 is set up for simultaneous EEG and pupillometry, or eye tracking, but pupillometry data collection. You have the sample experiment both in the Google Drive, and it's also down on the computer as well in a folder called iLink plus EEG. And so if you look through the walkthrough, it should give you everything you need to know how to customize. Go back to practical example 2. There's a recording of it posted now. And I go through in more detail how to customize that and work around the iLink code. Because the big thing here to know is the number of code chunks to make triggers go to the EEG system is so easy. So easy. The number of code chunks to make PsychoPy talk to the iLink is like, it's all over the script. So it's much easier to take the sample experiment that is PsychoPy talking to iLink and just slip in, okay, now send triggers to the EEG. That aspect is really easy, but I don't recommend reinventing the wheel, essentially taking an EEG experiment that you then need to add eye tracking to, just because the number of code chunks and putting them in the right places is going to be a lot to keep track of. So those are my recommendations. I would be thrilled if more people wanted to do simultaneous pupillometry and EEG here at BCBL, because we could troubleshoot this together and really get it going. I've already had some help with Manu. She has a black box which checks the timings, and so I was working on that in the last months. So, are there any questions? Yeah, Judith.
Because when I guess that when you show us the one example of data from iLink, the timing was a little bit odd, meaning that iLink started to initialize the time, like the row column timing weirdly, while the EG usually is very precise.
Yes.
So I guess I wonder how these two timings can work together, because I know that this is the only more problematic things when you have two systems to combine, is when the time series is not the same.
The same, yeah.
Luckily, I think that the iLink has the same sample rate than the EG, but sometimes not.
Yes.
I think that, for example, Toby doesn't have the same, so it's
yeah, it's a little complicated. Julia's asked an excellent question, which is how do we make sure the timing of the EEG and the iLink are the same? So if you remember in the data, when we look at the start of a data file that we've opened up in R, it starts at like 2,300,000, some crazy number, because iLink for every participant initializes at a different number, and I'm not really sure where the logic is behind it. But what we have and what we did in our tutorials yesterday is we always have the timing of the onset of our stimulus. So if our stimulus for trial 1 started at 2 million, we know that, and we actually renumber from 0. And so we will have, for our pupillometry data, we have from 0 for the start of each trial. When you go to your EEG system, even if the EEG system initialized on 6 million, which I don't think it does, but if it did, because we have the triggers sent to the EEG system, we also know the start of the stimulus for the EEG system's timing that has been recorded. Does that make sense? So for both systems, we have these key time points that are relative to that system. So we can line everything up, make the data essentially like normalize it as starting from 0 for each system, and then bring them together. That is my vision at the very least.
I have another million of questions.
Yeah.
Because I have the background with EEG.
Mhm.
That's with, but also, the question. I guess that then the other problem would be the artifact detection,
Yes.
because you have both artifact from the eye
Mhm.
that you have to control because it's also the predominant one,
Yeah.
and artifact from the brain.
Yeah.
And movement and, well,
Yes.
there are others.
Yeah, the artifact detection. And then, you know, what's interesting is Manu, who's our EEG expert here, for those who don't know, was telling me that there are ways to import pupillometry data into the EEG pre-processing steps, essentially like another channel. In which case you could do artifact detection in the same manner. And then also blinks will be corresponding with artifacts in the EEG signal. So it might be better to go about it that way. Because I come from the pupillometry background, I pictured processing everything in R like I always do, and then going and getting the EEG data and sticking it on it in R. But I think Manu would probably encourage me to do it the opposite direction and use some of the standardized EEG pre-processing.
Something like blinks, having eye tracking would actually help. You wouldn't have to do ICA potentially, because you could just, I mean, maybe you do it anyway, but you know where the blinks are.
Yeah, but the problem is that you have to find a way in which you have like to literally align your, like Manu said, but I'm working as a channel because you have to like AG, you detect the artifact also with a visual inspection. So if you have it perfectly aligned, you have to be sure to delete the same part in the EG and in the eye tracking part. If not, you will have a mess.
Yeah. Yeah, and so going back to that timing question, if iLink initializes at 2 million and some crazy number, with the R scripts that we've gone through, I very much line it up for each trial. But if you wanted the whole long pupil response and you wanted to line up the whole thing as a channel in the EEG system, you would need to probably look at the timing and figure out what it initializes on. Yeah, it's a good point.
As long as the sampling rate is the same.
It is, yeah. That's the other thing. I have set the sample. Yeah. Yeah, the sampling rate for this eye tracker that is downstairs in the EEG cabin can be 1000, and so it'll just always be set at 1000 in that room. So that at least removes one layer of difficulty that you would have with Toby. Yeah. Okay, I am going to wrap up there, and I'm going to set up for our lovely presenters to give